So today we're going to talk about immunoprophylaxis. What is prophylaxis basically? Prophylaxis are those methods which are being done to recover from a disease. In immunoprophylaxis, we imply that what all measures that we have to take to improve the quality of the immunity. And we know that the immunity of a person is mainly being improved by the presence of certain agents which will trigger the immune system. So by naturally getting exposed to an antigen, of course your immunity will be increased. But what are the other strategies by which you can improve your immunity? Of course, we have other methods like vaccinations, which we practice right now. And now we're going to talk about that and the history of vaccines updated. Let's see into it. So let's see about the vaccine development history. Okay, we know the immunoprophylaxis is mainly being done with the help of vaccines. And the first vaccine which we can trace down is the production of smallpox vaccine by Edward Jenner in 1796. And he noticed that the cowpox pustules, when they were being used for people, okay, when they were being infiltrated with cowpox pustules, what happened? The people developed immunity against smallpox. This discovery was mainly because of he serendipitously, that is by chance, he had observed that the milk ladies who were taking care of the cows having pox, okay, or those who are having cowpox, they were immune to smallpox, and that led to the development of Edward Jenner's smallpox vaccine. Later after that, you can see that the prominence of vaccination was there in Europe and as well as in US, and about 100,000 people got vaccinated in Europe during that time in 1801. And even Britain also passed a vaccination law in 1821 stating that all the working class people should get vaccinated. Hereafter, then in 1879, you should look how many years it took further on for a true vaccine to develop. Like cholera vaccine was being developed in 1879, and then we had Pasteur coming with the attenuated virus rabies vaccine. Also we got the tetanus and the diphtheria vaccines were being developed. So the contributions of Pasteur in the field of medicine is also there, but since it was Edward Jenner who started the first attempt in immunology, Edward Jenner is referred to as the father of immunology. And in 1916s we had the first large polio epidemic in US, and what happens then, they started searching for polio vaccines, okay, and in 1935, see, 1916 we had the epidemic, and in 1935 about 20,000 children were being vaccinated by field trials, okay. Okay, and then 9 of them died with during these field trials. Then after that we have the Salk vaccine. You know what is the Salk vaccine? The Salk vaccine is a polio vaccine itself, but it is not the oral one. It is the intravenous version of polio vaccine. And hereafter the Sabin vaccine comes. Sabin is the oral polio vaccine in 1961 to 1962. That time 1962 it was licensed, okay, and further on further developments were also being done. And further on, if you go to look, every year like different types of vaccines were being developed, okay. And oral polio vaccine was being developed in 1962, as we can see, okay. And then in 1974 MMR, mumps, measles and rubella vaccine was being developed. And then you have every different company was been investing, and some of the companies also had to run away from the vaccine industry because of the high cost that they had to spend over this. And then you can one important thing which I have to mention is Bill and Melinda Gates Foundation, okay. They launched grand challenges in global health initiative, and even now all the important strategies or important research activities which would lead to a societal benefit, that has been funded by the Bill and Melinda Gates Foundation. Okay, and now of course we know the situation. How is it? We are trying to find out vaccines against COVID-19, and how successful it will be, that depends upon the field trials, the first investigations, lab studies, then comes to the animal studies, and then finally we come to the clinical trials. So normally a vaccine, if it has to be developed, you have to give a uh, a lab proof saying that the vaccine is viable. Then after that you have to put it into animals and check whether any side effects are there on the animals. Then after that you go to the clinical trials. That is, in the clinical trials you will have human volunteers. That is, people itself, they will be willing to say that okay, you may inject me, I will be a experimental person where you can see. And then you check the efficiency of that vaccine in humans, and that these are referred to as the clinical trials. After that clinical trials in humans, then after that, once the results are validated, the vaccine is being allowed to be used on others. So normally it takes so many years to reach at that level of use of a vaccine. Okay, and now we do expect that efforts in the vaccine development against COVID-19 will overcome this barrier. Like rather than going into animal trials, we are not waiting, we are directly injecting them into the human trials. And how far it will be successful, as well as what are the side effects of it, it will vary. Now some milestones, as we had mentioned earlier, in 1940s we had smallpox. Okay, and also at that time, then after that DPT vaccine was being developed in 1940s, and 50s we had the smallpox and the polio vaccine, and late 1960s smallpox, DPT, as well as polio, oral OPV, measles, MMR, these were being detected. Okay, so I think these are clear to you. Now coming to again the history of the vaccine development, if we go to have, it's a different representation of this whole thing that we are saying. In the 18th century, in 1720, smallpox was prevalent, and the first successful vaccine, as we told, in 1796 was being developed by Edward Jenner. Then after that, different types of vaccines. You can see in 1855 we have the live attenuated vaccine, and then by 1886 we have the killed vaccine. The live attenuated rabies vaccine was used, okay, by Pasteur, and the killed vaccine was being used for cholera, plague, as well as for typhoid. Then the toxoid vaccines, that is a chemical inactivation. Okay, and so what is the thing that we have about live attenuated and killed? You know, live attenuated is something which the organism will be live, but its virulence will be reduced. Okay, and in killed vaccine, you kill the organism, and then that killed portions of the organism can be used as a vaccine. The next one that we saw, this is a toxoid vaccine. The toxins are being taken and they are being inactivated and they are being used. Then further we have the time of combination vaccines, where different combinations are being used. Then you can use the in vitro cell culture vaccines, that is, you take something and you will grow them in cell lines. Okay, then the polysaccharide vaccines, the glycoconjugate chemistry, the recombinant vaccines, the therapeutic vaccines, as well as the genome based vaccines. These are the history of the different types of vaccines. In the coming slides we will be learning about the different types of vaccines and what are their functions. Thank you for now.